To open the next session, please welcome Dr. Gloria Cristal-Luna, AOS2020 Conference President and past President of the Philippine Society of Medical Oncology, Philippine Society of Oncologists, and Molecular Oncology Society of the Philippines.
Good afternoon, ladies and gentlemen. The next lecture is entitled Deciphering Heterogeneity in Cancer, Big Data, Pathway, and Biology. Our distinguished speaker is Professor Rubiyon Ju Huang, Professor at the National Taiwan University, visiting professor at Kumamoto University, Japan, and Director of the Translational Center for Development and Research, Transcend National University Health System. She was a recipient of the Yushan Young Scholar Award from the Taiwan Ministry of Education. Ladies and gentlemen, please welcome Professor Rubiyon Ju Huang.
Hello, everyone. I am Professor Ruby Huang from National Taiwan University. First of all, I would like to thank the organizing committee for the invitation. Today, I would like to talk about how to decipher the complexity in cancers in the era of precision medicine by integrating three components: big data, pathways, and biology. As we know that there has been a significant paradigm shift in how we practice medicine. In the past, based on symptoms, we practice intuition medicine. As the outcomes resulting from our practice accumulate to form certain patterns, we entered the evidence-based medicine. Our practice is pretty much guided by different levels of evidence. With the development of technologies, many of these clinical information are present in the form of computation data that we would need to deploy algorithms to help us find those patterns. The future medical practice would therefore require the application of rules, algorithms, and database to enable actionable clinical decisions for tailored patient care. In a way, we have moved away from the one-size-fits-all medicine to the stratified medicine. In the past, we stratified patients based on disease types, demographics, or some clinical features and simple biomarkers. The future stratification strategy would further expand towards the individualized level to achieve the so-called precision or personalized medicine to incorporate many more parameters, such as the socioeconomic, environmental, behavior, and preferences, with much more complicated biomarkers. Nevertheless, for either stratified or personalized precision medicine, we would tease out the complexity by exploring three components: biology, data or big data, and pathway. These three components feed into each other and form a life circle journey which could be integrated into the patient journey, just like the Holy Trinity. Today, we will use tumor heterogeneity as the starting point of the biology in question here. The first layer of tumor heterogeneity usually refers to inter-tumor or inter-patient variations. Big data generated from the multi-omics levels, genomics, epigenomics, transcriptomics, proteomics, metabolomics, and microbiome has become readily available nowadays. The pathway is thus referred to how we use analytical tools to find common or differential patterns that could connect the variation networks or nodes embedded within the tumor heterogeneity. Let us take ovarian cancer as the example. This is a high-grade serous carcinoma. From the look of it, each tumor might look similar, but are they really? The management of ovarian cancer in the past followed stratification of histology. More recently, with our understanding of actionable mutations, the management of ovarian cancer has shifted to the stratification of mutations such as BRCA1, BRCA2, or homologous recombination repair genes to inform whether patients should be given PARP inhibitor treatment. However, even underneath the same mutation profile, ovarian cancer is still heterogeneous at the transcriptomic gene expression level. This is where big data comes into the picture. The transcriptomic data of high-grade serous carcinoma came from two landmark studies. The first study, Totil et al. In 2008, profiled 285 high-grade serous and high-grade endometrioid carcinoma from the Australian cohort. They concluded that there are 6 subgroups, C1 to C6, based on the gene expression profiles. A subsequent study from the TCGA cohort in 2011 profiled 489 high-grade serous carcinoma and concluded that there are 4 subgroups: differentiated, immunoreactive, mesenchymal, proliferative. To see whether these subgroups are reproducible, the meta-analysis performed by our group in 2013 pulled together 1,538 ovarian cancer, regardless of histologies, and identified 5 subtypes: Epi A, Epi B, which had better survival outcomes, mesenchymal stem A, which had worse outcome, and stem B. From this head-to-head comparison, you can appreciate how robust and concordant these subtypes are among these studies. This is a good sign for stratified or precision medicine, because we know that there is a potential to stratify patients based on these subtypes. Finding the pathway and network underneath these subtypes usually will shed light into the therapeutic development. In the example of ovarian cancer, TGF-beta pathway is found to be a significant node for the mesenchymal tumors. This would provide another actionable value for patient stratification, because now we can design how to treat these patients differently. So in a way, biology, big data, and pathway all feed into the center of patient stratification, with the end goal to inform the appropriate treatment strategy. This process highlights the need to develop diagnostics or companion diagnostics to make this happen. Let us go back to ovarian cancer. Based on the transcriptomic heterogeneity, ovarian cancer patients could have tumors conforming to good prognosis: the Epi A C3 differentiated, the Epi B C24 immunoreactive, the Stem B C6 subtypes, or tumors conferring to poor prognosis: the mesenchymal C1, the Stem A C5 proliferative subtypes. Efforts have been made to design diagnostic tools to stratify ovarian cancer patients based on these gene expression subtypes. The first assay is a 48-gene nanostring assay developed by Professor David Botel in Peter Mac, Melbourne. RNAs were extracted from the FFP sections of the tumor samples. You can see that they can robustly identify patients with this gene expression subtypes. More recently, a consortium effort developed a prototype assay, which also is based on the nanostring technology platform. With the development of diagnostic assays in hand, a clinical trial was launched in 2017 by our group in collaboration with Peter Mac to stratify ovarian cancer patients. In this case, the STEM-A C5 proliferated subtype to a treatment, in this case, the antimicrotubule vinorvin. To achieve this, a diagnostic workflow from patient recruitment to assay to report out was set up. I cannot show you the data since it is still ongoing, but I can share with you some caveats that we have learned. This protocol mandated 3 blocks from the tumor to be profiled. This is to ensure that we could have concordance within the tumor parts. Majority of the patients could be assigned to one subtype. However, you do encounter patients, such as this one, which show 3 different subtype calls from the 3 blocks. So what does it mean here? Let us go back to our Holy Trinity again. It turns out that there is a second layer of tumor heterogeneity that describes intra-tumor variations, meaning having differences within the same tumor. So how do we tackle this? We go back to the big data and pathway network again. We analyzed the gene expression profile of 3,431 ovarian cancer and realized that while 70% of the tumors were assigned into one subtype, approximately 30% were assigned as having more than one subtype. There was no mutual exclusiveness of how they were mixed, suggesting that there could be random mix and match. Would this affect the outcome? We first took those patients whose tumors were annotated with only one subtype and just looked into the intrinsic subclonal signals to reflect the heterogeneity. We developed a heterogeneity score to stratify these patients into high heterogeneity in red curve here and low heterogeneity in blue curve here. The survival curves tell us that for subtype with good prognosis such as APA or APB, patients with high subclonal heterogeneity actually did worse. So this tells us that the purer it is, the better the outcome. We next look at those having more than one subtypes. For these patients, it matters with whom you are mixed. We found that when the good prognosis subtype is mixed with poor prognosis subtype, the outcome is worse than just having the pure good prognosis subtype. The data also support that the worst case scenario would be the mixture of the 2 poor prognosis subtypes, mesenchymal and stem here, that the patient outcome is the worst. So as the Chinese saying goes, one really takes on the color of one's company. So what could be the pathway-driven biology we are looking at here? These are the two tumors with mixed subtype label. Please turn your attention to these red arrows, which indicates the presence of immune cells. The tumor were stained with CA125, an APA marker, and alpha-smooth muscle actin, a mesenchymal marker. You can see that inside the first tumor, which is EpiV stem A mixture, the alpha-smooth muscle actin did not extend inside the tumor mass. The infiltrating immune cells are very far from where alpha-smooth muscle actin is. However, for the second tumor, which is EpiB mesenchymal mixture, the alpha-smooth muscle actin was extensively dispersing within the tumor mass. The infiltrating immune cells in this case are trapped within the alpha-smooth muscle actin positive areas. If you are familiar with alpha-smooth muscle actin, this is a marker to label where myofibroblasts are, demarcating areas within the tumor where the extracellular matrix would be remodeled. So, would we expect the infiltrating immune cells in these two tumors to function the same? The answer is apparently no. Furthermore, would we expect these two patients to respond to the same, let's say, immune checkpoint inhibitors? In summary, I have illustrated to you that how the Holy Trinity, integrating biology, big data, pathway, could help us understand the complexity in tumor heterogeneity, both intertumor and intratumor. So what's next for the field? We are entering into the spatial biology domain to address tumor heterogeneity. A cross-disciplinary team integrating the oncology, pathology, and spatial genomics could lead us further into the frontiers of precision medicine. I want to leave you with a few questions to ponder upon. Can we stratify patients not by specific biomarkers, but just by heterogeneity itself? What is the neighborhood effect within the tumor? And how do we remodel the community? With this note, I would like to thank you for your attention.
Hello.
Hi. Good evening, Dr. Ruby Huang.
Good evening, Dr. Luna. How are you?
Thank you
Thanks for having me.
We're good. Thank you for joining us tonight.
Yeah, no problem.
Okay, can we move on? You have a very good presentation.
Thank
And
you very much.
I just hope everybody was able to comprehend as well as probably make use of this in the future or in the near future.
Yes.
So we have some questions for you from the audience.
Yes, sure.
And one of our participants would like to hear your answer. When we talk of heterogeneity in colorectal cancer, we also talk of consensus molecular subtyping or subgroups. And we often ask for tumor location. What about in ovarian cancer or in high-grade serous ovarian cancer? Is there a role for the cell of origin in determining the heterogeneity among the different tumor samples? And she said, when she say cell of origin, is it from the fimbrial epithelium or from the ovarian surface epithelium? or from the malignant ascites. Maybe
Yeah.
you can make use of this as a determining factor for the heterogeneity. Is there or is there none?
Yes,
But
there
because
is.
in colorectal cancer, we have differences between location. We can when the lesion is on the right or on the left, there are differences and different modes of treatment as well as prognosis.
Yes, thank you very much. This is a great question. Yes, cell of origin is a very important issue. We know that ovarian cancer derived from the fallopian tube is very different from the non-fallopian tube derived one, which is the ovarian surface epithelium. So the spatial heterogeneity is definitely there and is actually reflected by the histology. So the non-ovarian surface epithelium, some derived ovarian cancer, mainly these are the clear cell, these are the low-grade ovarian cancer, and we have the endometriosis associated with, which are the clear cell carcinoma. And we have the mucinous, which is very similar to the colorectal and gastric cancer. And this heterogeneity is basically at the very top of our categorization of ovarian cancer. We talk about histology in my talk. So, when you think of that, that is at the inter-tumor heterogeneity. And when you go into the intra-tumor heterogeneity, the spatial heterogeneity is also there, meaning that, like I said, which part of neighborhood are you in? I'm sure you can imagine it's like a metropolitan Manila. You would have a neighborhood that is more affluent, and you have some neighborhood that is more troublesome. I think it's the same for tumors.
Okay. If I might follow through, when we talk of heterogeneity, uh, we know that this is one of the mechanisms for drug resistance, right?
Yes.
So this exists within the individual tumor mutations. And in colorectal cancer, this is present in around 5 to 90% of sequencing. So the different heterogeneity here is expressed like in uh, seen as amplification, gain or loss, deletion, and so on. And in colorectal cancer again, we have this consensus molecular subgroups. Uh, and the prognostic value of this CMS classification could be confirmed, for instance, in the RAS wild type metastatic colorectal cancer, including the ORR. These are seen as beneficial. The question here is the CMS of colorectal cancer, like the immune, the canonical, metabolic, mesenchymal, is it different or similar to molecular subtypes of high-grade serous ovarian cancer? Which is your immune reactive, your differentiated, proliferative, and mesenchymal. Uh, Immune and the mesenchymal are almost the same for both. What about the canonical and metabolic? So, with regards distinct biological features and clinical outcome, how can we compare the two?
I think this is another great question. People, to keep the answer short, I think from some pan-cancer analysis, people have started to tease out, for example, what about the mesenchymal tumors? Are they similar? In short, they are enriched in signatures such as epithelial mesenchymal transition, the EMT transition. But you have to understand that the stroma compartment from ovary and from colorectal probably are very different. They probably have different resident immune cells. So how would the different organotropic immune cells versus the stroma compartment in the EMT pathway? That would be an interesting understanding. So that's why if you just look at the intertumor heterogeneity, the EMT mesenchymal, and you have to go inside the intra-tumor heterogeneity to look at basically the residents inside that geographic area? So that's a great question.
You have different classifications or subgroupings, no? Do you already have a consensus out of it?
Yes, I think
because you have C1 to C6, and then you have 4, and then you have 5.
Yes, I think for ovarian cancer community, we are sort of reaching that consensus. So that's why we have the head-to-head match for each other. So now we have a huge consortium trying to stratify patients using this consensus signature. So yes.
Okay.
Yeah.
So the novel histopath classification of high-grade serous ovarian cancer, plus the status of microenvironment, including the infiltration of immune cells, and stromal features. This corresponds to gene expression subtypes relevant to the prognosis and biological differences. And I hope this novel classification in the future could lead us to individualize our chemotherapeutic regimens for high-grade serous ovarian cancer.
Yes.
Do you agree?
Yes, of course. That's the aim.
So for your last message for our viewers, for our participants, can you give us a summary of what you have just given us, or lesson for today?
So the lesson for today is know thy neighbor. So like we want to have good neighborhood. I think when we treat the tumors, treat the cancers, we need to understand the entire tumor microenvironment, the cancer cell community. We have to understand we're not just treating the cancer cells, we're also treating their neighbors.
Yes. So nobody stands alone.
Correct.
We have to be cooperative with one another, just
Yes.
like One Asia.
Yes.
Okay. So if there are no more questions, I would like to thank Dr. Ruby or Professor Ruby for this particular knowledge learning, or we were able to differentiate now the different histopathologic findings in ovarian cancer.
Yes.
So
I hope you find it useful.
Serous ovarian cancer. Thank you very much, Dr.
Thank you, Dr. Luna. Thank you. Bye-bye.
Bye-bye.